This is going to be a short overview of cellular-derived inflammatory mediators. The first pathway that I'm going to describe is the cyclooxygenase pathway. This is one of the main pathways in cell-derived inflammatory mediators, and it's stimulated by either cytokines or bradykinin. These cytokines will bind to G protein-coupled receptors, which in canonical G protein-coupled receptor fashion will lead to the separation of PIP2 into IP3 and DAG, which will then go on to, in this specific case, activate MAPK. This MAP kinase will activate and phosphorylate phospholipase A2. Additionally, a secondary activator, in this case calcium, will be transported by calcium channels and will also activate phospholipase A2. As the name implies, phospholipase A2 will cleave arachidonic acid from the phospholipid bilayer. Arachidonic acid is one of the main central mediators of a bunch of these pathways, and so this is probably one of the most important molecules to know for the action of all of these pathways and the downstream effects that they have on the body. So in the cyclooxygenase pathway, arachidonic acid will then be converted into prostaglandin G2. These cyclooxygenases are targets of pharmaceuticals, and I'll go over that in a little bit more detail later. But prostaglandin G2 can also be then interconverted to prostaglandin H2. From here, prostaglandin H2 can be converted into a couple different prostaglandins, which have similar but a little bit different effects on the body. So the first one is prostaglandin I2, which is also known as prostacyclin, as well as thromboxane A2. Now, prostaglandin I2 is a pro-inflammatory mediator, which causes vasodilation and inhibits platelet aggregation. So I'm going to go ahead and draw that. In the center of my screen, I have all of the kind of downstream targets of these mediators. And so I'm going to go ahead and draw an arrow to signify prostaglandin I2 or prostacyclin causing vasodilation, as well as inhibiting platelet aggregation. Just going to go ahead and draw the little inhibitory marking. So it's going to inhibit platelet aggregation. Thromboxane A2, which is also derived from prostaglandin H2, is actually going to have the opposite effect. So it's going to activate vasoconstriction, so it's going to make the vessels constrict, and it's also going to increase platelet aggregation. There are two more prostaglandins that can be formed from PGH2. These are just the two that I'm going to mention. There are actually a few more prostaglandins that can be produced. But for simplicity's sake, I'm just going to talk about prostaglandin D2 and prostaglandin E2. Now, prostaglandin D2 is preferentially made by mast cells. It also causes the activation of downstream mast cells, so it plays an important role in allergy. But interestingly enough, prostaglandin D2 also causes vasodilation, but instead of altering platelets, it's actually going to increase vascular permeability. So that kind of makes sense when you think about how allergies work. You swell a lot when you have an allergy to something, thinking like hives or anaphylactic shock. You swell, right? So vasodilation and vascular permeability makes sense in that case. Prostaglandin E2 is the main mediator of pain and fever, and obviously fever is one of the main ways the body fights against infection. So this is part of the innate immune system. It's part of making an environment inhospitable for pathogens. So this is kind of an overview of the cyclooxygenase pathway. Now, in a similar light, I'm going to talk about the lipooxygenase pathway, which is similar, but it has a little bit of a different flavor. So lipooxygenase pathway can happen, in addition to the cyclooxygenase pathway, can happen in a wide variety of leukocytes. So in this side of my screen, I've drawn just a generic leukocyte. This is also stimulated by some sort of cytokine, whether that be IL-1, TNF, some other cytokines that are, floating around in the plasma. And this will have a similar effect as before, which will activate phospholipase A2, which will create arachidonic acid. But now, rather than being the reaction being catalyzed by cyclooxygenases, this reaction is actually going to be catalyzed by lipooxygenase 5. And that's going to create 5-HPETE. This 5-HPETE can then also be converted to 5-HETE. In addition to 5-HPETE, we can actually get our first leukotriene, so leukotriene A4. Now, leukotriene A4 is kind of another intermediate in this pathway, and we can go down a couple different steps here. So the first possibility is having an interconversion to leukotriene B4. We can also have the creation of leukotriene C4, D4, and E4. Or we can have an alternate pathway, which I'll go over in a little bit. So the first ones I want to bring to your attention in terms of what they do to the body is 5-HETE and leukotriene B4. These two mediators actually act mostly on neutrophils. So these two mediators will cause neutrophil chemotaxis as well as neutrophil activation. So I'm going to put chemotaxis and activation. Alternatively, our other leukotrienes, leukotriene C4, D4, and E4, induce bronchoconstriction and vasoconstriction and increase vascular permeability. So these are also major players in allergies and asthma causing bronchoconstriction. I just wanted to kind of draw that out here. We're going to draw a little arrow showing that these three leukotrienes cause bronchoconstriction, or the closing of the bronchi within the respiratory system, as well as vasoconstriction. And they're also going to increase vascular permeability. Alternatively, leukotriene A4 can actually then be further modified in a separate mechanism of the lipooxygenase pathway, which will create lipoxins. So LTA4 can be catalyzed by 12 lipooxygenase, as well as lipoxin A4 and lipoxin B4 hydrolase to create lipoxin A4 and lipoxin B4. And now what's interesting about this specific pathway is it's actually a transcellular production pathway. And so in order for lipoxins to be created, you actually need the interaction of the leukocyte and the platelet. So this pathway can't specifically happen just in the leukocyte. And so what's interesting about the lipoxins is actually it inhibits neutrophil adhesion and chemotaxis. So it kind of has the opposite effect of 5-HETE and leukotriene B4. So I'm going to go ahead and draw that out. This is going to inhibit neutrophil adhesion and activation. So just for clarity, I'm going to write out inhibits adhesion. So that is the lipooxygenase pathway. It's similar, it has similar mediators to the cyclooxygenase pathway, but then the end results are a little bit different, and they have a little bit of a different effect on the body, specifically the downstream leukotrienes causing bronchoconstriction. So the next pathway I'm going to go over is the platelet activating factor pathway. This pathway can happen in a wide variety of cells, as I've drawn here. It can happen in neutrophils, leukocytes, endothelial cells, as well as platelets, but it happens in actually a very similar way. It's kind of a side pathway to the cyclooxygenase and lipooxygenase pathways. So, as a byproduct of arachidonic acid synthesis, we actually get the production of lyso-PAF, or lysoplatelet activating factor. And after the conversion of lyso-PAF by LPA-LAT, we actually get platelet activating factor. And as the name implies, this can activate platelets. And so it actually enhances platelet aggregation as well as activating leukocytes in contributing to the synthesis of reactive oxygen species. So I'm actually going to draw a little bit of a cycle arrow here. Showing that although PAF can be created by all of these specific cells, it can also activate those cells. And additionally, I'm going to draw an arrow up to our little platelet aggregation diagram, showing that it does enhance platelet aggregation. So the last pathway that I am going to describe is the nitric oxide pathway. Now, this pathway is a little bit different in the case that it's not really lipid mediated, though it is produced by cells, a couple different cells with a couple different flavors of the specific enzyme that creates nitric oxide. So in macrophages and other leukocytes, in other phagocytes, we have inducible nitric oxide synthase. And so this is actually inducible by cytokines and other stimulants in the surrounding area to these specific cells. And this specific enzyme catalyzes the creation of nitric oxide from L-arginine. And so there's kind of two effects that this nitric oxide can have on the body. If it's near a blood vessel, it can actually mediate paracrine signaling by causing vasodilation, as nitric oxide usually does. But in the more direct sense, phagocyte-produced nitric oxide actually can contribute to the creation of downstream reactive oxygen species, which can actually act on pathogens and cause them to lyse. So this is actually part of the pathway where phagocytes can actually kill pathogens in the body as part of the innate immune system. Additionally, in endothelial cells, we have endothelial nitric oxide synthase, or eNOS. This also synthesizes nitric oxide from L-arginine in the endothelial cells specifically, and can cause vasodilation. It also can cause smooth muscle cell relaxation, which really contributes to vasodilation. So the last point I want to make is just a couple drugs that were mentioned that can inhibit some of these pathways and have impacts on their action and kind of mediate the mechanism of action of these specific drugs. So the first drug that I wanted to mention is glucocorticoids. And glucocorticoids mainly inhibit phospholipase A2. They can also inhibit the cyclooxygenases. Additionally, NSAIDs, or non-steroidal anti-inflammatory drugs, mainly inhibit the cyclooxygenases. And so what this does is that it actually decreases the production of some thromboxanes and prostaglandins. They can lower fever. It can also lower pain. Albeit that the glucocorticoids, what you should know is that they're broader acting. And these are so the NSAIDs are a bit more specific. So those are two drugs that were mentioned that play into this pathway. They also have effects on some of the enzymes in the other pathways, but I just wanted to point those out as a little aside to the main impact of the pathway. Without further ado, I wanted to introduce the last little part of this video, which is the secretion of histamine and serotonin. Now, these are mostly secreted by mast cells, and these can also, this specific release of these vesicles is really as a result of cytokine activation. That can be IL-1, IL-8, as well as any anaphylotoxin. So as we talked about before, that can be C3A and C5A from the complement system. There actually are a couple others that can activate mast cells, but these are mainly mediators of allergy and inflammation. And so as a result of the cytokine stimulation, you get release of histamine from mast cells. It can also be released from platelets and basophils, but I just, for simplicity, just stuck to mast cells. And so if you think about mast cells being mediators of allergy, they actually can contribute to the symptoms that you associate with allergy. So that would be swelling, your throat closing, and that's in severe cases of anaphylactic shock. But these actually, the downstream physiological effects of histamine makes a lot of sense in this instance. So histamine actually causes bronchoconstriction. That's when you feel your bronchi are closing, your throat might tighten a little bit when you're in anaphylactic shock, as well as vasodilation and an increase in vascular permeability, and that leads to the swelling that you see in allergies. So overall, I hope this gives you a good overview of the different parts of cell-derived inflammatory mediators. Please let me know if you have any questions. Thanks.